to be going over chapter 10, blood. Now you are going to see that the main function of blood is going to be to transport substances throughout our entire body. That is going to be from head to toe. Now what is it that is going to be carried within blood? Pretty simple. You're going to have nutrients, vitamins, minerals. We are going to also be carrying waste such as urea, uric acid, carbon dioxide, carbonic acid, hormones. These hormones are going to be very important for our endocrine system, as well as that you're going to see that blood is going to be capable of transporting body heat. That is to allow us to keep us warm. Now remember, blood, it is a type of connective tissue. And again, remember, why is it considered a connective tissue? It is because it's going to connect your entire body. Again, that is going to be from head to toe. Now, of course, blood is going to be composed of different components. We're going to have our living cells, and we're going to go over those in a second, but these include red blood cells, white blood cells, as well as non-fluid matrix, such as water. Now, what happens if you separate blood? Now, first and foremost, how is it that we're going to be capable of separating blood? Now, if you look into your slide number 5, you're going to see that blood can be placed into a centrifuge. What is a centrifuge going to do? This is going to get a sample of blood in the test tube, spin it multiple times, and you are going to see that the heaviest substances within blood are going to drop down, while the lightest are going to remain on the top. Now, once blood is separated, you're going to see that it's going to be divided into 3 different sections. We're going to have the very bottom section where you're going to find all of your erythrocytes. Now, you're going to see that erythrocytes are going to compose 45% of your total blood, and this is going to be referred as hematocrit. Your second layer of blood, after being separated, we're going to refer it as the buffy coat. And this is going to be where we're going to find our leukocytes and platelets. And finally, we're going to have our last layer, which is going to compose of plasma. Now, plasma composes 55% of your blood, and this is going to be where you're going to find most of your water as well as other substances. Again, slide number 5 here gives you a better illustration of how it is that blood is going to be separated. Remember, our lowest layer is going to be referred as hematocrit, and this is going to be where we're going to find 45% of our entire red blood cells. We're going to have our middle layer that is going to be composed of our white blood cells, and we're going to refer this middle layer as our buffy coat, and our top layer we're going to refer it as plasma. And again, within plasma we're going to find mainly water, but we're also going to find salts, electrolytes such as sodium, potassium, calcium, chloride, so on and so forth, and we're also going to find proteins. So before we go into what we're going to find in each of these layers of blood, you're going to have to understand the blood characteristics. How is it that we can distinguish blood from any other fluid in our body? Now, first and foremost, remember, blood does have a distinct color, but we're going to have a range of color. We are going to be capable of finding blood in a scarlet reddish color, and that is going to be when blood is going to be highly oxygenated, but we can also find blood in a kind of bluish color, and that is going to be when blood is deoxygenated, meaning that there is going to be very little or few oxygen molecules within it. Now, you're also going to see that blood is going to be much heavier and thicker than water, so it's going to be much more dense. Also, you are going to see that blood will have a metallic kind of salty taste. And why is this going to be the case? Because remember, within blood we are going to find iron. Iron, it is a metal. Especially if we have high concentrations of iron in our blood, it's going to be much more a metallic taste compared to when we're going to have high concentrations of salt. That is going to be either sodium chloride. Then we're going to have a more of a salty taste. Now, one thing that is going to be very important for you guys to remember, and that this is not going to be the last time we're going to see, is going to be the blood pH. Now, the blood pH will have a range between 7.35 and 7.45. Again, that is going to be slightly, again, remember, if we go back to the chemistry chapter, a solution that is in close proximity to a pH of 1 or below 7 is said to be acidic, while a solution that is above 7, close to 14, is going to be said to be alkaline or basic. Now, another characteristic of blood is going to be its temperature. Now, how much blood is found in our body? We are going to find about 5 to 6 liters of blood within a healthy adult. Now, of course, there are going to be conditions out there and diseases out there that could increase the volume of blood or can perhaps decrease the volume of blood. Nonetheless, in a normal, healthy individual, quote unquote, normal individual, we're going to see about 6 quarts. All right, so let's go ahead and start with the first layer of blood after it has been centrifuged. And this layer, remember, this is going to be the highest layer of the 3, which we're going to refer to as plasma. Plasma is going to be composed of 90% water. But in addition to that water, we are going to find solutes within it. These solutes will include nutrients, electrolytes, again, all those vitamins, all those minerals, your sodium, potassium, magnesium. We're also going to find respiratory gases such as carbon dioxide and oxygen. We are going to find hormones. We're going to find plasma proteins as well as waste products. Now, what are going to be the type of proteins that we're going to find in plasma? Now, you're going to see that most of these proteins are going to be synthesized by the liver. And one of these proteins includes albumin. Now, what is the function of albumin? Now, in later chapters, we're going to see how albumin works in conjunction with your cardiovascular system to maintain normal blood pressure. Another type of protein that we're going to find in plasma include clotting proteins. And remember, these proteins are going to be very important, especially when we are trying to heal a wound to prevent any blood loss when any vessel has been damaged. And the third type and very important type of protein that we're going to find in plasma include antibodies. And remember, these antibodies are going to be the ones responsible to help you fight any type of infection and create an immune response to those pathogens. You are going to see that the composition of blood and plasma will vary depending on the need of the body. Now, for example, if you are suffering from some sort of bacterial infection, of course, the percentage of antibodies are going to be much more higher compared to an individual that is not fighting any infection. But do note that the respiratory and the urinary system are going to work together with the cardiovascular system to maintain the proper blood pH. Remember that blood pH, normal ranges of blood pH are from 7.35 all the way to 7.45. Anything above the 7.45, your blood is going to be too alkaline. If your pH drops below the 7.35, then your blood is going to be too acidic. That does include even if your blood pH is at 7. Now, in this case, in this scenario, yes, a pH of 7 is considered neutral, but when we are talking about blood, if your blood drops to a 7, it's going to be too acidic. Now, what are going to be the type of foreign elements or living cells that we're going to find in our blood? We're going to find erythrocytes, which can also be referred as red blood cells. We're going to find leukocytes, can also be referred as white blood cells, and we're going to also find platelets. Platelets are going to be only cell fragments, are not full cell. Now let's start with the erythrocytes, or your red blood cells, abbreviated RBCs. Now, what is going to be the function of your erythrocytes? Again, going back to the general function of blood, it is going to be to carry oxygen, to carry carbon dioxide in and out of your tissues. Now, the function of erythrocytes is going to be to actually carry the oxygen around through your entire body. Now, some characteristics of erythrocytes compared to other cells, including your white blood cells, you are going to see that red blood cells are anucleate, meaning they do not contain a nucleus. They will contain few organelles and they will lack mitochondria. And because they are anucleate and they contain few organelles, the red blood cell's lifespan is going to be very short compared to the lifespan of other cells that do contain organelles and do contain a nucleus. However, your red blood cells are going to be again very essential and very important to carry a protein. This protein is hemoglobin. Now, I will be going over hemoglobin in a second, but you are going to see that red blood cells will have a very distinct shape, and that shape is going to be a biconcave shape. Now, what is a normal count of red blood cells in your system? Now, you're going to find 5 million red blood cells per cubic millimeter of blood. Now, how much is that? Now, think about a regular drop of blood. Cut that drop into 50 equal parts. Each of those 50 parts should contain those 5 million red blood cells. And so as you can see, our body containing 6 quarts or anywhere between 5 to 6 liters of blood will definitely contain a lot of red blood cells. Again, and these red blood cells will compose 45% of your hematocrit. Now remember, within your red blood cells, we're going to find one very important protein, and this protein is going to be your hemoglobin protein. Now, what is going to be the function of hemoglobin? Again, remember what is the function, what is one of the functions of blood, is going to be to carry oxygen around. Now, this protein, this hemoglobin protein, is the one responsible to carry the oxygens throughout your system. Now, you're going to see that each hemoglobin protein is going to be composed of 4 subunits or 4 smaller proteins. Each of these 4 smaller proteins within the hemoglobin protein will be capable of binding to one oxygen molecule, which means that per one hemoglobin protein, we're going to carry 4 oxygen molecules. And so within each erythrocyte, we're going to find 250 million hemoglobin molecules, which means that approximately within one erythrocyte, we're going to find 1 billion oxygen molecules. Now, if we want to be a little bit more precise, we're going to see that normal blood will contain anywhere between 12 to 18 grams of hemoglobins per 100 mLs of blood. And again, remember that is going to be a high volume when we remember that a normal healthy individual will contain anywhere between 5 to 6 liters of blood. Now, one homeostatic imbalance of the red blood cell is going to be anemia. So let's just describe and define an anemia. So anemia is going to be the decrease of oxygen carrying ability of the blood. Now, yes, note that there are different types of anemia. For example, we're going to have hemolytic anemia. We're going to have iron deficiency anemia. We're going to have hemorrhagic anemia. But nonetheless, regardless of which anemia you're talking about, which anemia you're describing, they're all going to share the same condition or symptom. That is, again, the decrease in oxygen carrying ability, which means that if you suffer from an anemia, any type of anemia, you're not going to have proper levels of oxygen. Again, remember, what is your percentage of hematocrit? That is going to be 45%. If at any time that percentage of hematocrit decreases dramatically, let's say 30%, 35%, what does that mean? It means that you're not going to have enough red blood cells. If you do not have enough red blood cells, you're not going to have enough hemoglobin proteins. If you do not have enough hemoglobin proteins, you're not going to be carrying enough oxygen. If you do not have enough oxygen, your cells and your tissues are not going to be capable of receiving the proper levels of oxygen, which means that they're not going to be capable of functioning properly. Now, again, if we want to be more specific about anemia, anemia could be due because of a lower than normal number of red blood cells, such as the example that I gave you with a decreased hematocrit. But in addition to a decreased hematocrit, we can also suffer an anemia due to abnormal or deficient hemoglobin content in the red blood cells. So one, if we do not have enough red blood cells, we will not have enough oxygen. If we have enough red blood cells, but not enough hemoglobin proteins, we're again not going to have enough oxygen. Now, one specific example of a type of anemia is sickle cell anemia. Now, this is going to be the result from an abnormally shaped hemoglobin, such as the example in your bottom right. And again, this is going to be the result from a mutation in the protein of hemoglobin. Because of this mutation, the hemoglobin is not going to fold properly. It's going to make the cell have a sickled shape. Again, because it has a sickled shape, the hemoglobin is not going to work properly, which means that it's not going to be capable of carrying enough oxygen, which means that your tissues and or organs are not going to receive enough oxygen either. Now, slide 15 will give you a table about the different types of anemia. And again, regardless of the type of anemia, remember they all going to share the same characteristic, which means that they're going to be incapable of carrying enough oxygen to your cells, to your organs, to your tissues. Now, another homeostatic imbalance of your red blood cells include a disorder referred as polycythemia. Again, if we take this term itself and break it into smaller pieces, we're going to have poly, which means multiple, and then we're going to have cythemia. Emia refers to your red blood cell. So what is polycythemia? It's going to be the disorder that will result in excessive amounts of red blood cells. Now there's going to be two specific types of polycythemia that we're going to be shortly describing. That is going to be polycythemia vera and secondary polycythemia. Now, is polycythemia good? Is polycythemia bad? You're going to see that polycythemia can be good, but will definitely be bad in the long run. How is that so? So let's take, for example, polycythemia vera. Now, polycythemia vera is bone marrow cancer. Remember, cancer is an abnormal cell growth reproduction. Remember, what happens if we have too many cells in our blood is going to be way thick. If it's too thick, it's not going to be capable of flowing through our vessels. If it's incapable of flowing through our vessels, healthy red blood cells are not going to be capable of flowing through our vessels, which means that oxygen will never reach those organs and or tissues. So as you can see, polycythemia vera is bad. Now, how is it that polycythemia can be good? Again, we're going to have, for example, secondary polycythemia. But even though I'm telling you right now that secondary polycythemia can be good, if you are exposed or you have secondary polycythemia for a prolonged period of time, it can result once again in too thick of a blood, which will result in your organs and or tissues not receiving the adequate supply of oxygen. Now let me explain how it is that having polycythemia for a short period of time can be a good thing. Now you're going to see that if you live at high altitudes, your body will automatically respond to that high altitudes. How? By synthesizing more red blood cells. Now, why is it synthesizing more red blood cells? It is because at high altitudes, you are going to find lower concentrations of oxygen. So once living at high altitudes, your body is going to compensate the lack of oxygen in the environment by producing more red blood cells. Now that your body has more red blood cells, it's going to be capable of binding to more oxygen molecules, which means that your tissues and or organs are going to receive the proper levels of oxygen at all times. But remember, if you live at high altitudes and move to a higher altitude place and then move to an even higher altitude place, what is your body going to do? It's going to continue synthesizing red blood cells. And remember, this increase in red blood cells will eventually slow your blood flow, which means that your blood is not going to be capable of running properly or flowing properly through your vessels, which means again that your system, that your organs and or tissues are not going to receive the adequate supply of blood. All right, so now let's move to our leukocytes. Again, these can also be referred as your white blood cells. Now, what is going to be the general function of your white blood cells? It is going to be to protect your body and serve as a defense against microorganisms, against pathogens that might want to invade your body and make you sick. Now, compared to the red blood cells, leukocytes are going to be complete cells, which means that they will have organelles, they will have nucleus. Now, of course, the number of leukocytes are going to be much less compared to your red blood cells. Now, why is that the case? That is because your leukocytes are going to become activated and they're going to be needed only if an infection or a foreign microorganism tries to invade your body. Now, you are going to see, however, that your leukocytes are going to move around your entire body, also from head to toe, but only when needed. And they're going to move through a process called diaperesis. And this is going to happen when chemicals are released from damaged cells. These chemicals are going to call upon the white blood cells. Now the white blood cells are going to go to the damaged or injured area. And this process is referred as positive chemotaxis. Now, what is the normal white blood cell count in a quote-unquote normal healthy individual? You're going to find anywhere between 4,800 to 10,800 white blood cells per cubic millimeter of blood. So from now to the end of the leukocyte section, you are going to be responsible to interpret values given you, such as interpreting what it means if an individual has, for example, 12,000 white blood cells per cubic millimeter of blood. In that example, remember, a quote-unquote normal healthy individual will have a maximum of 10,800 white blood cells per cubic millimeter of blood. So what happens if this individual has 12,000 white blood cells? It probably means that its body is producing more than normal white blood cells in response to an infection. So again, you are going to be responsible to interpret any values that I will be giving you. Now, you guys do not need to memorize the normal values. I will be giving you the normal ranges. And again, all you have to do is to tell me what that value means. For example, leukocytosis. This is going to be the condition that is going to happen when your white blood cell count is above the 11,000 cells per cubic millimeters of blood. And again, this high levels, higher than normal levels of white blood cells, will generally indicate an infection. What kind of infection? That does depend on the type of white blood cell that is going to be elevated. Now, what happens in the other hand if we have lower than 4,800 white blood cells per cubic millimeter of blood? Then we're going to undergo leukopenia. Again, this is abnormally low white blood cell count. Now, why would a person have leukopenia? Now, you're going to see that some diseases, such as cancer, after treatment for cancer, drugs will usually inhibit your immune system, which means that it's going to decrease your white blood cell count. Now, what does this mean to the individual? Because this individual will have low levels or low counts of white blood cells, they are going to be an easy target to pathogens, as the body is not going to be capable of responding accordingly because they're not going to have enough white blood cells to fight that infection. Now, what happens, however, if the white blood cells become cancerous, then this is going to be referred as leukemia, bone marrow cancer. Now, in leukemia, we are still going to be producing white blood cells, but these white blood cells that are being produced are going to be immature white blood cells, which means that they are going to be there, but they're not going to be functional. And because they are not functional, the body is still going to be sensitive to pathogens and or microorganisms and make the person ill. All right, so we have gone over leukocytes. Remember, this is just going to be the general term that we're going to give to the white blood cells. Now we're going to go a little bit more into detail into the different type of white blood cells. Now, these white blood cells can be categorized as either granulocytes or agranulocytes. Now, basically, all I need you guys to know is the list of granulocytes and the list of agranulocytes. In granulocytes, we're going to have neutrophils, eosinophils, and basophils. And they're going to be categorized as granulocytes because if you were to look into one of these leukocytes, you're going to find a bunch of little granules or dots or specks within the cell itself. Now, the agranulocytes include lymphocytes and monocytes, and these will lack those little granules that you're going to find in the granulocytes. Now, here's a list of white blood cells from most to least abundant. Now, the most abundant white blood cell is going to be the neutrophils, followed by the lymphocytes, then the monocytes, eosinophils, and finally the least abundant white blood cell are going to be the basophil. Now, I have included a mnemonic that will hopefully help you remember this list quite easily, and that is by remembering never let monkey eat bananas. Neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Again, from most to least abundant. So let's go ahead and start with the neutrophils. And again, remember, these neutrophils are going to be the most abundant white blood cell that we're going to find compared to the other type of white blood cells. Now, if we go into the normal values or normal ranges of neutrophils, you're going to find anywhere between 3,000 and 7,000 neutrophils per cubic millimeters of blood. Again, these are normal ranges. So what would it mean if we have elevated neutrophils counts? Let's say, for example, 8,000 or 9,000. Because we do have elevated number of neutrophils, it's going to automatically tell us that your body is trying to fight some type of infection. Now, you are also going to see that the neutrophils are going to be your first responders. What does that mean? So let's say, for example, that you have a deep paper cut in your hand. As soon as you have that deep paper cut in your hand, neutrophils are going to be called upon the injury site, and the neutrophils, with its phagocytic function, is going to try to prevent any pathogens from entering the wound to infect your entire body. Again, the neutrophils are going to be your first responders. Now we go back to the values. What would happen to an individual if the neutrophil value is below the 3,000? Let's say, for example, 1,000, 1,500. It means that if at any time the person suffers from an infection, the body is going to be incapable of fighting off that infection. Again, just to reiterate what I mentioned before, you are going to be responsible to interpret values. I will be giving you normal ranges. All you have to tell me is whether the value that I give you is too high or too low and what that value means. The next type of leukocyte that we're going to be talking about are eosinophils. Now the normal ranges, the normal values of eosinophils are going to be anywhere between 100 and 400 eosinophils per cubic millimeter of blood. Now what is going to be the function of eosinophils? Eosinophils' function is going to be to kill parasitic worms. And they will also play a role in allergic attacks. So let me give you another example. You have a patient that has no visible wound. However, after blood samples were taken and results were delivered to you, you see that their eosinophil counts are at 500 eosinophils per cubic millimeter of blood. What does that mean? It means that this person, this individual, has a parasitic worm. Remember, eosinophil can tell us if a parasite is present in the individual. Now, you are going to see also that whenever you do suffer from an allergic attack, let's say, for example, right now that flowers are releasing too much pollen on the environment, if you go outside and you start sneezing, you are sneezing because there's going to be high levels of eosinophils trying to prevent that pollen from entering your body. Now remember, eosinophils can help you determine whether a patient has a parasitic worm or perhaps is just suffering from an allergic attack. The next type of white blood cells are going to be the basophils. Now the normal ranges of basophils are going to be anywhere between 20 to 50 basophils per cubic millimeter of blood. Now, what do I want you guys to remember about basophils? Now, basophils, they are also going to work with your eosinophils in allergic attacks, but most importantly, basophils will contain heparin. Heparin is an anticoagulant. Now, what is going to be the function of basophils in an opened wound? Basophils are going to go to the opened wound and is going to prevent blood from coagulating too much. That is going to prevent the blood flow to the damaged area, which will allow neutrophils, for example, to do their function, which is going to be, again, to kill and or prevent any pathogens from entering your body. All right, now we're going to go into the agranulocytes. And the first agranulocyte that we're going to be talking about are going to be your lymphocytes. Now, what is the normal values of lymphocytes? These are going to be anywhere between 1,500 to 3,000 lymphocytes per cubic millimeter of blood. Now, these lymphocytes will play a major role in your immune response. Why is this going to be the case? It is because lymphocytes are going to be the ones responsible in synthesizing antibodies. Now, for now, this is all you have to remember about lymphocytes. Once we go into the lymphatic system, we're going to go a little bit more into detail into how it is that antibodies are going to be synthesized by these lymphocytes. And finally, we're going to go over the monocytes. Now, monocytes' normal values will be anywhere between 100 and 700 monocytes per cubic millimeter of blood. Now, what is going to be the function of monocytes? Now, these monocytes are going to be released in a chronic infection. So that means when you have been fighting an infection for a prolonged period of time, perhaps 2 weeks. So if your lab results come back with monocytes values of 900 monocytes per cubic millimeter of blood, what does that mean? It probably means that you have been fighting an infection for quite a long time now. And finally, we're going to go over the last type of formed element that we're going to find in our blood. and that is going to be the platelets. Remember, platelets are only going to be cell fragments from another type of cell, and this cell is going to be a megakaryocyte. So what's going to happen? Megakaryocytes are going to grow and develop until they reach maturity. Once they are mature, they're going to release these fragments called platelets. And so these platelets are going to be very important for the clotting process during a damaged tissue. Now you're going to find a normal platelet count of 300,000 platelets per cubic millimeter of blood. Now again, remember, you're going to be responsible to interpret values. So what does it mean if your platelet count is above the 300,000 platelets per cubic millimeter? It probably means that somewhere around your body, whether exposed to the environment or internally, your body is trying to fix something where blood is coming out. Perhaps a ruptured vessel, a ruptured capillary. Now, slide 27 all the way to 29 will give you once again a table that is going to give you characteristics of every single cell that we have previously discussed, from erythrocytes to your white blood cells, including neutrophils, eosinophils, basophils, lymphocytes, monocytes, and your platelets. Once again, you guys are not going to be required to memorize the normal ranges, the normal values of all of these cells. However, you will be responsible in interpreting values. All right, so in this next section, we're going to see how it is that blood cells are going to be formed. Now, this process is going to be referred as hematopoiesis. Now, where are blood cells going to be formed? This is going to occur in the red bone marrow. Now, remember, the red bone marrow can be found in long bones, in the middle portion of long bones. Now, you are going to see that every single blood cell is going to be derived from a common stem cell. This is going to be the hemocytoblast. Now, slide 31 will give you a better illustration of how it is that every single blood cell is going to be formed. Again, the hemocytoblast is going to create either a lymphoid stem cell or a myeloid stem cell. The lymphoid stem cell is going to produce the lymphocytes, and the myeloid stem cell is going to produce either the erythrocytes, the basophils, eosinophils, neutrophils, monocytes, and the megakaryocytes that are going to create the platelets. Now remember, red blood cells will have very specific characteristics. Understand that red blood cells are going to be anucleate, which means that they're going to be incapable of dividing, growing, and synthesizing proteins, which is why the lifespan of a red blood cell will be anywhere between 100 to 120 days. However, because your body is constantly using red blood cells, your red bone marrow is going to be also constantly producing these so that you can replace those damaged or worn out red blood cells. Now you're going to see that your spleen and the liver are going to remove these damaged red blood cells, or these red blood cells are no longer useful. Now, how is it that red blood cells are going to be produced? Now here we're going to once again go back to the endocrine system. A hormone is going to tell your red bone marrow to start producing these red blood cells. So what's going to happen? If there is an abnormally low concentration of oxygen in your body, your kidney is going to recognize that low concentration. In response, your kidney is going to release the erythropoietin hormone. This erythropoietin hormone is going to target your bone. Your bone, in response, is going to synthesize more red blood cells. And this is going to make sure that your body is kept in homeostasis regarding oxygen levels. Now, what prompts your white blood cell and platelet formation? Once again, we're going to have more hormones. Now, here we're going to have the release of chemicals. Now, the colony stimulating factors and interleukin chemicals are going to prompt your bone marrow to generate the leukocytes. And the thrombopoietin hormone is going to stimulate the creation of the megakaryocytes, which in response going to synthesize the platelets. So let's say, for example, that you do have a cut in your finger. Those cells that have been damaged are going to start secreting the colony stimulating factors and interleukins, which are going to go directly into your bone, telling your bone to start the production of white blood cells and platelets. Now, hemostasis is going to be the process of stopping bleeding whenever a vessel has been broken. This is going to happen, of course, to stop and prevent any hemorrhage. Now, hemostasis is going to happen in 3 phases. We're going to have vascular spasm, platelet plug formation, and coagulation. So what is the first step of hemostasis? Again, this is going to be vascular spasms. And what does that mean? So vascular refers to the actual blood vessel, while spasms refers to contraction. So in vascular spasm, your vessel is going to immediately respond by contracting as much as possible so that the damaged vessel can close as tight as possible to prevent any further blood loss. Step number 2, platelet plug formation. Again, remember, these cells that have been damaged are going to start to secrete chemicals such as your thrombopoietin hormone. Remember, this hormone is going to make sure that there are going to be adequate levels of platelets present in your body. So what's going to happen? Platelets are going to rush themselves into the damaged site, creating a platelet plug or a white thrombus. And so what this plug is going to do is going to prevent, once again, is going to prevent any further blood loss. And so step number 3 is going to be the coagulation process. It is going to be the step before your blood vessels start healing. And so what's going to happen? Your cells are going to start secreting chemicals such as tissue factors and PF3, which is a phospholipid. Now, all of these chemicals are going to start to trigger the clotting cascade. And so what is this clotting cascade going to do? It's going to convert prothrombin to thrombin so that thrombin can interact with fibrin. And now these two, fibrin and thrombin, are going to be capable of creating a meshwork, which is going to create a more stable clot so that you completely prevent hemorrhage or blood loss. Again, here in this illustration, we can see how it is that thrombin and fibrin join together to create a more stable meshwork to completely prevent blood loss. Now, the process of hemostasis is going to happen anytime within 3 to 6 minutes, which means that approximately by minute 6 after you have damaged a blood vessel, there is going to be no further blood loss. And so within these 6 minutes, step 1, step 2, and step 3 of hemostasis are going to happen. Now remember, after the tissue has been fully repaired, then this clot is going to deteriorate once again so that everything goes back to normal. Now there are, however, some disorders of hemostasis. We can potentially undergo undesirable clotting or blood clots. For example, a thrombus. A thrombus is going to happen whenever the clotting process starts spontaneously. So it's going to happen even though there is no damaged vessel. Now, of course, this can be deadly, especially if a thrombus blocks blood flow to a very important organ, such as the lungs, heart, or the brain. Now, an embolus is going to happen whenever a thrombus breaks away, floats around the bloodstream. Now, once again, this can most definitely prevent blood from going to tissues and or organs. Another type of hemostasis disorder is going to be the bleeding disorder, such as thrombocytopenia. Now, this is going to happen whenever there is insufficient numbers of platelets in your body. And remember, what is the function of platelets is going to be to start the process of preventing blood loss. Now, if a person does suffer from thrombocytopenia, what can happen? Even with the smallest cut, they can bleed out, they can go into a hemorrhagic shock, which means that they're going to be losing high volumes of blood, which could perhaps lead to a deadly situation. Now, evidence of thrombocytopenia is going to be petechiae, which is going to be small blotches or even small dots in your skin that happen because of ruptured capillaries. Now, hemophilia is a hereditary bleeding disorder. Now, similar concept as thrombocytopenia. Now, in hemophilia, there is not going to be normal clotting factors, which means that your body is going to be incapable of clotting correctly, which perhaps can lead to hemorrhage because there is going to be no way to stop the bleeding. Now, if at any time a person loses anywhere between 15 to 30% of their blood, this is going to automatically cause weakness. Why is that? It is going to be because your vessels are not going to be capable of transporting enough blood to organs, and tissues are not going to receive adequate supply of oxygen, which means that there's not going to be enough energy, which means that you're going to feel tired. Now, if your body loses over 30% of blood, this will definitely cause a shock, which, if not treated fast, it will lead to death. Now, in order for us to treat blood losses, we're going to have to understand how transfusions are going to occur and how it is that a transfusion can save a person's life. So, for example, if you do suffer from anemia, through a transfusion, the new blood is going to be fully oxygenated, which means that your organs and tissues are going to be fully oxygenated, which means that everything is going to work as they should. Now, in order for us to understand blood transfusions and who can donate and who can receive, we have to first understand how blood groups and blood typing works. Now, this information you guys are going to find in the second video. Now, this second video is going to be part of your lab for this week. So make sure you completely understand how it is that blood typing works. And just as a heads up, there is going to be a quiz on blood typing by the end of the week.